Okay, welcome back to this explanation, and we have meiosis second here as a equational division where it's similar to mitosis as well, and it separates sister chromatids. Each haploid cell from meiosis 1 divides again. So every phase again here it's called second, okay? It's the same names, but just what is different is the number. Instead of be meiosis or prophase 1 is meiosis second and prophase second. Okay, here, getting closer, we have a prophase second as a chromosome recondenses again, nuclear membrane breaks down again, and new spindle fibers form. The chromosome numbers are haploid and chromosomes still duplicated. So nuclear membrane breaks down again and the spindle fibers begin forming in each cell. As you can see, there is 2 cells because that was the result from meiosis first. Okay, here in metaphase we have chromosomes line up individually at the metaphase plate. Each sister chromatid is attached to spindle fibers from opposite poles. Okay, nuclear membrane completely absent and spindle fibers attached to chromatids. Following for the anaphase second, the sister chromatids finally separate. They move to the opposite poles and chromosomes numbers, the chromatids are separated. Each become a chromosome. Then we have the telophase second phase, where the chromosomes reach poles, clear membranes reform and around each group, and the chromosomes decondense. As you can see, everything happened at the same time in each cell. Here, the telophase, the cells start to elongate because that will result in a separation into individual cells having another pair of cells, resulting in 4 daughter cells. Okay, now we have a little comparison about meiosis first and meiosis second. Well, meiosis first reduces the chromosomes numbers from diploid to haploid, and the meiosis second chromosomes numbers stays haploid in the whole process, and the nuclear membrane disappears or breaks down during prophase number 1 and then reform on telophase first. In meiosis second, nuclear membrane breaks down again in prophase second and reformed in telophase second. The spindle fibers attach to homologous chromosomes, and during meiosis first, by other hand, in meiosis second, spindle fibers attach to sister chromatids. Crossing over only occurs on meiosis first, on prophase, and is not happening here during this meiosis second. The diversity of the genetic genes and the crossing process, it just happened during the meiosis first. Something important to remember is that meiosis prophase first in females happen when they are an embryo inside of the uterus of the mother, and it stops there, and meiosis restart when the female has the first period. But in another hand, for males, after puberty, meiosis starts and happen every day. creating the eggs and the eggs for the women, as I said, and the sperm for the males happen every day. So this was my presentation, a little bit I think a little bit long, but thank you for listening.